Alright, welcome back everyone. I hope everybody has had a good week so far and a great weekend. Today we'll be covering chapter 5. We're going to continue that um I guess lectures that's covering from the general sense of biology into more the molecular um, aspect of biology. So today we're going to be covering the structure and function of large biological molecules. Okay, so we're gonna go over large molecules like carbohydrates, fats, proteins, and nucleic acid at the end. Okay, all living things are basically made up of these four large biological molecules. Um, from the food that we eat, from the actual organisms, from plants. You know, we're talking about like big organisms all the way to the micro cellular atoms. They're just made up of these biological molecules. And so when we say macromolecules, is basically macro means a large, right? So we're talking about large molecules. Okay. When we say micro, that's small molecules. Okay. So large molecules basically have unique properties, and those unique properties are based on the way they look, the way um, they function, the way they have their structure is maintained together. It's three-dimensional shapes. Uh, it's arrangement of atoms around itself and around other molecules that allows it to interact with other molecules around it. This is an animation of a structure of protein and its function. You guys can watch this on your own. So large polymers, as the name states, it's basically a molecule that's large. And basically, polymers is a long molecule that's consisting of many building blocks. Okay, those blocks we call monomers. So monomers are small, um, basically chains. Okay, that when you put them together, we come up with a polymer. Okay, and a polymer that are basically large enough, we call them macromolecules. Again, that word that's describing a large molecule. Okay. Uh, the carbohydrates, proteins, and nucleic acids are polymers. Remember, so we're gonna talk about four categories today. So we're gonna talk about carbohydrates, proteins, and nucleic acids, and fats. So you can see that fats are not listed as polymers. So they're not polymers. Okay. So we're gonna also just briefly describe, what enzymes are. Okay, I'm just going by how the book basically is listing these terms. Okay, so enzymes are basically proteins made out of proteins. Okay, and they're specialized macromolecules. Okay, so you see that term again. And their specific jobs are to speed up chemical reactions that basically make or break down polymers. Okay, so they speed up the chemical reaction, but they do not add or subtract from that chemical reaction. So they're not. Do you remember that term reactants that we learned about from before in the chemical equation? So they're not reacting in an actual chemical reaction. Okay, they're just like helping to speed it up. So a dehydration reaction, when we say a dehydration reaction, occurs when basically two monomers, remember those small blocks that make up a polymer, right? When those two monomers bond together, right, and there's a water molecule that's been lost due to that. Okay, that we call this a dehydration reaction. So an example, and we're gonna probably like you're gonna see several figures in a bit that's gonna show you basically a lot of examples and how basically everything is put together. So an example would be like when we put together, for example, two amino acids, okay, to create a peptide chain, okay. Or another example would be like when we put two simple sugars together to create a disaccharide or a starch, okay, or a glycogen chain. We're gonna talk about all these terms in detail in a bit, so so you're not confused about this stuff. Okay, so polymers are basically the other way. So we talk about how to put it together, but how to break it apart. So if you have a polymer, right, a full chain of monomers, the way we break it down is we use a process called hydrolysis. Okay, so you can see that it's divided into kind of two sections: hydro and lysis. Lysis means you break apart. Okay, and hydro means it refers to water. Okay, so that's how we can do that. Okay, and a reaction that is essentially the reverse of the dehydration reaction. So it's exactly the opposite of dehydration. So this is an example showing you how dehydration reaction happens, how a hydrolysis reaction happens. Okay. So a dehydration, right? When we have that, we have a short polymer or a what? A monomer. Okay. And we have another monomer as well. And basically, what happened is uh, the hydrogen is and on one monomer reacts with the hydroxide on the second monomer. Okay, and when they do, basically, water is being basically formed. Okay, so that's removed out of the equation as dehydration occurs. Okay, and the two molecules join together and become longer polymer. Okay, or a polypolymer. Okay, so hydrolysis is exactly the opposite. So you start with a long chain, and we're trying to lyse it or break it apart. Okay, so when we try to break down that into two, that's when we add water molecule. Okay, so remember dehydration, water is basically removed. Hydrolysis, water is basically added. Okay, so it's added to be able to uh, split apart these, into two monomers. Okay. So all the videos, animated videos, you can watch on your own. You have the PowerPoint available, so I'm not gonna waste time going over it right now. They're pretty simple for you guys to watch and and go over things. So we're gonna talk about diversity of polymers. So a typical cell of an organism has thousands of different macromolecules. You can imagine it's got proteins, carbs, fats, nucleic acids, and nucleic acids are basically what makes up a DNA. And that's located in the nucleus of a cell, and that's what codes for everything. Okay. Macromolecules vary among cells of an organism, right? You're not going to have the same type of, you know, macromolecule in one organism than another. You might have um, more macromolecules in a certain species than another, okay, or a different type of that. So, um, this is just a brief description of carbs. So carbohydrates include sugars, right? And the polymers of sugars. Okay. So when we say sugars, technically we're referring to simple carbohydrates, okay, or a monosaccharide, okay. Now, when we make a large molecule of a carbohydrate, such as glycogen or starch or things like that, we're going to talk about that. We refer to these as polysaccharides. Okay, they're composed of many sugar building blocks or um, monosaccharides. Okay, now the simplest carbohydrates are monosaccharides. Okay, so they're the simplest um, monomers, right, in a as a carb. Okay, so you can imagine that everything in the body that the body consumes from the diet, like you know, carbohydrates, proteins, fats, in order for the body to absorb anything from the diet, it has to break down everything to the smallest units of monomers, okay, to be able to absorb it. It cannot really absorb large molecules the way it is without breaking them apart. So you can see how important um, is to learn about, you know, the simplicity of these molecules and the complexity of them. Okay. So let's talk about monosaccharides, or the simplest type of carbohydrate. So they have a molecular formulas, sorry, that are usually multiples of CH2s with an oxygen attached to it, all right? So in the form of glucose, that's an example, that's the main example actually of monosaccharide. We can see that we have 6 carbons, 12 hydrogens, and 6 oxygen, okay? And this is probably one of the most important monosaccharide carb that's you know, really important for the body and even for the plant and so forth. Okay, so the monosaccharides are classified. There's a classification for them. So there's we can look at the location of the carbonyl group. Okay, now whether it's an aldose or ketose. Remember, we have from previous chapter we talked about, you know, how um, the classification of monosaccharides can change depending on if it's an aldose or a ketose. Do you remember those two? So also the monosaccharides are classified by the number of carbons, right? The more carbons a monosaccharide has, the longer the chain is gonna be. Okay. When we talk about chain, it's basically the backbone of of a hydrocarbon, right? And carbohydrates are hydrocarbons. Okay. So this is what when we say aldoses and ketoses. Okay. These are the types of sugars. You can see here a depiction of them. Okay. And since we talked about glucose, glucose falls under an aldose. Okay. And for the purpose of us, you know, talking about all of this stuff, just focus on glucose mostly. Okay. Um, galactose is another form that's important, and fructose, which is the most type of sugar, simple sugar found in fruits. Okay. Now, as far as the body, which one the body absorbs normally as the simplest type of sugar after you consume a carbohydrate, that would be a glucose. Okay, so the body uses glucose as its blood sugar. Just notice here also the double chain, the double bond, and its location. Okay, you see if it's located, you know, at the end right here or in the middle, you see how it changes um, the type of sugar. So we go from an aldose to a ketone, okay, which is a ketose. Although we still have that 3 carbon, it's still the same. You see that? So where that double bond is located will change the dramatically the type of monomer we have, okay? And this is a pentose. Pentose is it means it's coming from the word pento or 5. Okay, so that's a 5 carbon chain. See that? And you can notice the location of the double bond as well. Same with these hexoses, so like glucose. Okay. And I want you to notice also this purple part. Look how similar they both are the glucose and the galactose. They're very similar. Even that double bond chain is also the same location. You notice the only difference is the purple part. It just flipped around where the hydroxide is facing this on the right side and the hydroxide on the galactose is facing the left side. And just that small change, it actually means a lot and does big differences in absorption and types of sugar that we actually have. So, although you see them like, drawn as a linear skeletons, right? As we saw in these, right? They're actually, these sugars are normally in aqueous solutions, like in water and so forth. They're normally formed as rings instead of just straight chains, okay? As we talked about monosaccharides such as glucose, they actually serve as a major and very important fuel for cells and raw material for building molecules. So not only that the cells can use them for fuel, but cells also, after the body consumes um, the amino acids, which is the breakdown of protein, and you know glucose, which is a breakdown of carbs, and just simple fats. The body sometimes wants to build back those simple molecules into complex molecules, okay, for whatever it needs, okay. If it wants to build, for example, a hormone, or it wants to build back some muscle that's been damaged, all of that it requires energy. And you can see that these monosaccharides can actually serve as energy and raw material to build molecules. Okay, so this is just a depiction of how these linear monosaccharide chain, they basically, in aqueous solution, what happened is they wrap around each other and they form a ring such as what you see here. Okay, and the way this happens is normally between the oxygen and the carbon. Okay, do you remember that electronegativity we talked about? This is where this comes in right here. Okay, and this is basically what you have as a result. Okay, sorry. So a disaccharide, as the sound, right? Di is two. So it is formed when we have a dehydration. What is dehydration again? It's when we put two monomers or two monosaccharides together, right? And what happened is water is basically what is excreted, okay? Out basically. And that's when we can form a disaccharide, okay? And we form a bond between the disaccharide or the two monosaccharides, and that would be what we call a glycosidic linkage. Okay, let's look at this. Hopefully, yep. So you can see here in this dehydration reaction, okay, the synthesis of maltose, and that's a disaccharide. Okay, so we can see how a glucose wants to join another glucose, which is another monosaccharide, and how when they do join and they form this 1,4 glycosidic linkage, as you can see here, you can see how water is basically excreted out. Okay. Same thing with when we try to form sucrose, and guess what? Sucrose is actually table sugar, right? So our table sugar is actually not a monosaccharide; it's actually a disaccharide. So it's not really the simplest form of sugar. Okay? So the way we form it is when we add glucose and fructose together, and these terms are very similar, so you just pay attention to those terms. Okay? Also water is being formed when we add them together. Okay? and we get that glycosidic linkage. You guys can watch also this animation on your own. So polysaccharides, as the sound, right, of it, it means we are forming a poly chain, okay, of many monosaccharides, not just two, but many of them, okay. We form that for storage and structural roles. So storage, such as starch or glycogen. Glycogen is basically the form of the form that the body uses to store carbohydrates and basically in your muscles and liver and so forth. Okay, the architecture and function of a polysaccharide are determined obviously by its sugar monomers, right? And the position of glycosidic linkages. So you can imagine by now that all of these molecules are very, very sensitive of the position of their molecules themselves, the position of where those functional groups are, right? The side chains, where those glycosidic linkages are. All of that is very important because they're really a puzzle. So the moment you change one thing, it's gonna really affect the rest of the chain. Okay. Starch, as I mentioned, is storage of polysaccharide of plants. Okay. We store as humans carbohydrates in the form of glycogen. Plants store it in the form of starch. Okay? And here we go. So glycogen is storage of polysaccharide in animals. Okay? It's stored mainly in the liver and muscle cells, as mentioned. Hydrolysis of glycogen, which is basically the breakdown of the big complex carbohydrate molecule, will release obviously those small simple glucose monosaccharides that the body basically demand for fuel. Okay. So this is just a depiction. It's actually in the book. This is showing you the polysaccharides between the plants and animals. Okay, so you can see how complex those chains are. Okay, in the form like this is starch right here. Okay, that's found basically in the plants. Okay, so the body basically needs, not the body, I mean, but whenever the plants wants to use that starch, it needs to break down those molecules into monosaccharides. Okay, same thing with the glycogen. Okay, and one difference that I need to mention is the difference between glycogen chains. Okay, that's stored in animals and humans. Okay, they're usually branched. Okay. However, when we look at cellulose, which is basically the form of carbohydrate that comes from plant cell, okay, and it basically surrounds the cell wall, okay, and these cellulose molecules are unbranched. That's important, okay. And you can see that the unbranched, because they're unbranched, they can form hydrogen bonds in between their chains. Okay. Due to this, they become very difficult to digest. Okay. So the polysaccharide cellulose, and this is what we're talking about here, the plant cell wall, okay, is a major component, right, of the tough wall of plant cells. Like starch, right, cellulose is a polymer of glucose, okay, same thing. This is what we're talking about. We're talking about a complex chain of carbohydrates, okay. But the glycosidic linkages differ, and they differ, those glycosidic linkages, they differ because we have a branched here, and we have an unbranched chain here. Okay. Chitin is another structural polysaccharide, okay, and that's found in the exoskeleton of arthropods. Okay, what do you think, that will do? It's basically a tough structure that protects those arthropods. Okay, so it also provides structural support for cell walls of many fungi. Okay. All right, so we're gonna talk about lipids right now. So lipids are basically a class of large molecules, right, that are not included as true polymers. Do you remember that from the beginning? So we said that carbs, proteins, and nucleic acids are all polymers, okay, but lipids are not, okay. And as we know from before, uh, lipids, due to their um, nonpolar bonds, okay, they are, they get away from water, so they don't mix with water at all, okay. They consist mostly of hydrocarbon regions. Hydrocarbons means what? Carbons and hydrogens, right? The most biologically important lipids are basically fats, right? Phospholipids and steroids. These are all made out of fats, mainly, okay? And we will see how steroids are actually made specifically from cholesterol as their main foundation molecule. Okay, fats are consisted of two types of smaller molecules. One is called glycerol, and another one is called fatty acids. And you will see that usually we have a simple single glycerol bonded to several fatty acid chains or hydrocarbons. Okay. A fatty acid consists of a carboxyl group basically attached to a long carbon skeleton, as I mentioned. Usually this is a hydrocarbon chain. Alright, we will see how that looks like in the next slide. A glycerol, however, is basically a 3-carbon alcohol, right, with a hydroxyl group attached to each carbon. Okay? So the glycerol part of this molecule will allow it to bond to other molecules. Okay, and this looks like a lot of things, but we'll look at it little by little. Okay, so um, fats basically separate from water because the water molecules hydrogen bond to each other and exclude the fat. Okay, so fat is not hydrophilic; it's hydrophobic, and it has those nonpolar bonds, and because they're nonpolars, they're not gonna dissociate in water. Okay. Um, in fats, 3 fatty acids are basically joined to glycerol, as we mentioned before, right? By an ester linkage, creating a triglycerol or triglyceride. Okay, that ester linkage you can see here. So this is basically the fatty acid chain. That's another one, and that's another one. Okay, you can see the ester linkage in between them, and that's the glycerol, by the way, that section right here. Okay. Now, when we formed what happened? What are we doing exactly? We are doing a dehydration reaction, remember? What happened because of that? Water is excreted out. As a result of this, an ester linkage is formed, okay? And what's cool is that we can form actually a fat that's consistent of different fatty acid chains length. So it doesn't have to be exactly the same or exactly the same length. You can see that these two are kind of similar, but the third one, this one in the middle, is different. However, it's still part of this whole um, fatty acid. Okay. So as you saw, right, these fatty acids vary in length, right, depending on how many carbons in the chain, and if there is also a double bond, or if it doesn't, it doesn't have to have a double bond, right? Now, saturated fatty acids basically have the maximum number of hydrogens, right? Do you remember from our chemistry lecture? Usually when there is a double bond, what happened to those hydrogens that are occupying the carbons, right? They're not there anymore, right? Because the electrons are being shared more between the carbons, right? You have a carbon here and a carbon here, and you have a double bond now instead of a single bond. Okay? So what happened to those hydrogens? They're not there. They can't bond anymore. The carbon is allowed to have how many? 4 bonds total. Okay? So unsaturated fats have one or more double bonds. Okay, and we'll talk about the difference between saturated and unsaturated fat. And these are important things for our everyday diet, by the way. If you look at any label of a food that you buy, you will see those two terms, saturated fats and unsaturated fats, and they're important for health and for many reasons. Okay. I wanted to mention this briefly before I move on. So what you're seeing here, that's a butter. Okay. We can see the difference between butter and that's olive oil. Why this is called saturated fat, okay, and why this one right here is called unsaturated fat, okay, and why this is healthier than this, okay. So what you notice right away is that the saturated fat is is basically like straight chain of hydrocarbon molecule, while the unsaturated fatty acid, it has a little kink here. You see that? And that kink is due to this addition of a double bond, basically. So this double bond happens to be in a cis configuration, and as we learned from before, a cis configuration means that the hydrogens are what on the same side of that three-dimensional molecule. Okay? And so when that happens, it creates that kink in the molecule. Okay? And that kink is basically what allows olive oil to remain actually liquid at room temperature. Okay? That the absence of that kink here in this saturated fat is actually why butter is actually solid at room temperature, and it needs to be heated up to become liquid. Okay? So, this is exactly what we were talking about, right? So fats made from saturated fats, okay, they're solid at room temperature. The fats that are made from unsaturated fats, like olive oil, they're liquid at room temperature, okay? So most animal fats are saturated. Plant fats and fish fats, right? Fish fat would be what? Omega-3. Have you heard of that before? I'm hoping you've heard about that before. So they're usually unsaturated. Okay. What would you think? The major function of fat is basically storage of energy. So whether it's an animal or human or whatever, right? They create fat specifically for emergency fuel. Okay, so when the body is not consuming carbs or sugars, it breaks down fat to get that energy over. Okay, So as you can see here, humans and mammals, they store them in cells, what we call adipose cells or fat cells. Okay, and these adipose tissue, not only they provide long energy storage, but they actually insulate the vital organs like the heart, stomach, um, intestines, kidneys. They actually have a layer around them that allows them to be protected and cushioned and not just be like solid. They actually move around and protects them from any type of like shock or temperature changes as well. Okay, so let's talk about phospholipids a little bit, right? Phospholipids are also types of fats, right? What you're seeing here is basically the model of a phospholipid. You're seeing in this case right here, two hydrocarbon chains. You can see that, right? So phospholipid has two fatty acids, okay, and a phosphate group. See that phosphate group right here? Do you remember from our previous chapter, a phosphate group is basically a functional group that's attached to a carbon chain. Do you remember that? And the phosphate group is basically a phosphate bonded to four oxygens. Okay? So that phosphate group is also attached to glycerol. Okay? Anytime we talk about fats, just in general, we're talking about glycerol with fatty acids attached to it. Okay? Phospholipid has that phosphate added right here. Okay? And it has a choline as well. Okay? Choline has basically an amine right here, and all of this basically there's a reason why a phospholipid is bonded to phosphate and choline, and that will make it capable of bonding to water. Okay, so it creates that hydrophilic or able to bond to water, right? Hydrophilic head. This portion of it is a hydrophobic, or it cannot bond to water. It cannot be broken apart by water. Okay. This is the depiction of the phospholipid bilayer. Where do you think this is located? This is actually basically what surrounds all of our cells. That's the membrane of our cell. Okay. You can imagine that things on the outside, okay, can enter the cells, but on certain conditions. So this phospholipid bilayer prevents the cell from prevents many things from entering the cell that are not allowed. Okay, we will talk about all of these things like at later chapters. Okay, so you can see that the heads are hydrophilic on both sides, the inner side of the cell and the outer side, while the hydrophobic tails are facing inside. Okay, so when phospholipids are added to water, right, they assemble into double-layered sheets called bilayers, as you can see here, right? This is how they are in all the cell membranes. You cannot see them just like a single, uh, a single phospholipid layer, because this wouldn't be like biologically, allowable. Basically, the cell cannot function like that. It needs to have that hydrophilic heads on the exterior of the cell and the interior of the cell. Okay. So steroids. Steroids are basically also made out of fats, mainly specifically cholesterol. I mentioned this, I think, a while back. So this is the how cholesterol looks like. That's the molecule of it. Okay, you can see it's got 4 carbon rings. Okay, attached to a side chain. Okay. This cholesterol is basically the precursor of most of our hormones: testosterone, estradiol, and different type of the hormones that the body uses. Okay. All right. So we're gonna talk about proteins now. So we talked about carbs, we talked about fats, and now we're gonna talk about proteins. So proteins account for more than 50% of the dry mass of most cells. That's a huge number. So you can imagine how important they are. Okay, some proteins speed up chemical reactions. What are we talking about here? We're talking about enzymes specifically. Do you remember that from the beginning of this lecture? Enzymes, when we say enzymes, they're actually proteins. Okay. Other protein functions, they include obviously defense. What are we talking about defense? Talking about the immune system cells. Okay. And their antibodies the antibodies that immune cells produce to fight viruses and bacteria. Okay, they're made out of proteins. Um, you can find them in storage molecules. You can find them in transport, cellular communication, movement. Okay, all of that. When we say cellular communication, we're talking about mainly what? Hormones, right? Structural support. Okay. Um, we will see how they actually play a vital role in uh, structurally support a cell. Okay, and they play a major role during cell division. We'll talk about all of this stuff in future chapters. Okay, so this is just a little example of each, right? So enzymatic proteins. Okay, you can see how an enzyme depicted here, you know, as a purple. Okay, they accelerate the chemical reaction, so it speeds it up. Okay, in this case, digestive enzymes, right, like proteases, for example, they catalyze the hydrolysis of bonds in food molecules. Okay, so so they're basically breaking apart those bonds and speeding up reactions. Okay, defensive proteins, I talked about that. So antibodies that help the cell fight viruses. These are made out of proteins. Storage proteins, right? In the case of casein, that's a type of protein in milk, and it's a major source of amino acids for baby mammals. Okay, and that's where protein is stored. Okay, they can be transport proteins. We will study a little bit later that you remember that's a cell membrane, but bilayer, phospholipid bilayer. These are basically transport protein that actually sit as a gate on the phospholipid bilayer, and basically they open and close to allow certain things in the cell and outside the cell. Okay, hormonal proteins are basically um, how the body communicates really. So insulin, for example, right, is secreted when there's a high amount of blood sugar. So insulin is secreted, and insulin brings down the blood sugar back to normal. Okay. Receptor proteins. Okay. That's in the case, for example, um, between muscles and nerves. Okay. See that? Contractile and motor proteins, right? Muscles are made out of proteins, right? Structural proteins, again, things like connective tissue, things that allow the cell to maintain their shape. Okay, collagen is very very important for tendons, for for your joints, like in the knees and shoulders and elbows. Okay. So again, enzymes are proteins that act as catalysts. They speed up chemical reactions, okay? They carry out those functions without actually participating in the chemical reaction, okay? Proteins are constructed from basically 20 amino acids. So when we're talking about protein in generals, we're talking about like, you know, polypeptide chains. So we're talking about several um, amino acids, which are the simplest form of proteins. Okay, when they come together, they become a polypeptide. Okay, in between the polypeptides, you know, we find peptide bonds, and these peptide bonds is basically how amino acids come together, and they form that polypeptide chain. Okay. So protein, as you can imagine, consists of one or more polypeptides, just depending on what type of protein we're talking about. Okay, amino acids are organic molecules with amino and carboxyl group, and we talked about this briefly last chapter, right? We have an amino group. Do you remember that? The NH2 as a functional group, right? Bonded to an alpha carbon. Okay, and this chain can obviously be like longer, not just two carbons. This is just an example. Okay. Amino acids differ in their properties, right? Due to the differing side chains called R groups. Okay. So we will see how when this R group changes, we will have different type of amino acid, and that different type of amino acid is vital, right? It's important in carrying out different functions and forming different polypeptide chains of proteins. And here is your carboxyl group. Do you remember that carboxyl group from previous chapter? I'm not gonna go over all of this. Just take a look at this just briefly. These are your 20 amino acids um, that form polypeptides. Okay, you can notice just the side chains, that R group I was talking about here. You can see it. You can see the differences here, right? If there's only an H, we have this type of amino acid, glycine. If it's bonded to more chains of carbons or other things, like a ring or so on, it just is forming a different type of amino acid. Okay. What you notice is the fact that some amino acids are actually hydrophilic while others are actually hydrophobic. Okay. Uh, these are hydrophobic. Do you see that all of these hydrocarbon chains, okay, these are hydrophobics. But when we add other things like oxygens and amine groups and so forth, we create the ability to be hydrophilic, or we can disassociate in water. Okay. What you notice is also that some amino acids, okay, are actually ion, okay, ionically charged. Some of them act as acidic, okay, if they're negatively charged. You see that oxygen, how it's negatively charged. Some of them act as basic, okay. Normally, if you remember from the previous chapter, if you have uh, uh, a carboxylic at the end as a side chain, like in this, right? You're gonna be what? Acidic. If you have an amine group, you're gonna be what? Basic. Do you see that? And that's how the different amino acids are basically differentiated. So amino acids are basically linked by covalent bonds. And these bonds are called peptide bonds. We will see hopefully here an example in a bit, okay? Polypeptide is basically a polymer of amino acids, okay? So it's made out of many amino acids. That's why it's called poly, okay? Polypeptide is many amino acids. And they range obviously in length, depending on how many amino acids, is being used. Each polypeptide has a unique linear sequence of amino acids, right, with two sides. We have a carboxyl end, or C-terminus, that's what we call it, and we have an amino and N-terminus. And we'll see in the next slide right here what that means, okay? So you can see here that between this left side right here is an amino acid, okay? And we can see on this side is another amino acid, okay? We can see how they can form this peptide bond in between, okay? And we can see that when the way they form that is through a dehydration reaction. Again, when we're putting two things together, you're doing what? A dehydration. So thus water is being released. Okay? You can see that we're forming a polypeptide with different amino acids that have different side chains or an R group. So when we do that, we're basically creating a new molecule that does a different job. Okay? You can see that the backbone is here. Okay? And we have a amino end. You see how that NH2 amino end? This is what we call N-terminus. On the other side, we have a carboxyl end. Do you see that? This carbon bonded to a hydroxide and an oxygen. This is what we call the C-terminus. So the beginning and the end, okay? Again, like anything we talked about, the way these proteins are formed and the way their three-dimensional architecture is, that's what's going to determine their function, okay? They can be twisted, they can be folded, they can be coiled, we will see how the different structure actually of proteins differ. There are simple ones, there are more complex ones, and this is why we're here. We're trying to learn about proteins and different things, right? So this is an example of basically an antibody protein. That's a defensive antibody, right? It's made out of protein. You can see how a protein from flu virus, that's just an example how it's like a puzzle. Remember how I told you that's a puzzle? So the three-dimensional shape of this is actually made specifically by your immune cells, right? According to the three-dimensional structure of the antigen, right? The word antigen means it's a molecule being secreted by the virus that the body recognizes, and basically they form the antibody according to that. And when it forms that puzzle like a lock, right? And that lock will allow the antibody to disable the virus or basically attack it, okay? So there are different levels of structure of proteins, as I mentioned before, yeah? You have your primary structure, which is the simplest form of basically a chain of amino acids linked together. Then you have your secondary structure, which is basically it's still a single chain, but however it's like twisted together, like you see here in this helix, okay, and this coil, I mean, okay. and it's still, remember, it's still a single chain. Okay. Sometimes that chain can form around each other like in a coil, and sometimes it forms a beta plated sheet. Okay. And we will see, like, we'll talk about the differences between each structure in the next few slides. Okay, we'll dig deeper into that. A third one will be tertiary structure. So now we're talking about more complex, right? We're talking about more three-dimensional. Now we have basically several chains, right, of amino acids interacting with each other, creating this tertiary structure. Okay, and we can see that the side chains, or R groups. Remember, we talked about that a few slides ago. These basically are responsible for this like three-dimensional shape. They bond with each other. Um, one R group from one chain bonds to another R group from another chain, and they form this three-dimensional tertiary structure. A fourth one would be quaternary structure, and this is basically when a protein consists of multiple polypeptide chains. Okay, we'll talk about each one in a bit. Okay, so let's talk about let's see here. So we're talking about the tertiary. This is just a depiction of a tertiary stabilization or a structure of protein. Okay, so in the tertiary, as I mentioned, structure, there is interaction um, and a bond formation between different chains of amino acids. Okay, and these different interactions, right? They are formed by hydrogen bonds. Okay, and they're formed by hydrogen bonds in the case of the polar amino acids. Remember how there's polar ones or hydrophilic ones and hydrophobic? Okay, so the hydrophilic ones will form hydrogen bonds. The hydrophobic um, side chains, like we see here, when they're close to each other, they form a hydrophobic interactions, and we call that van der Waal interaction. So remember, van der Waal is formed between the hydrophobic, okay, or nonpolar R groups, while the hydrogen bonds is being formed between the hydrophilic ones. Okay. Now, we also have ionic bond, right? Remember how some amino acids are actually ionically charged, like this one? Do you remember what's this is act that's this amino acid is acting as what? As an acid, right? And this one is acting as what? A base, right? It's got an amine group, and this one's got a carboxyl group. So that bond in between them, they're both charged. So what do you think is they're gonna form? An ionic bond, right? And then some of them have sulfur, like cysteine, I believe, right? Cysteine, if we go back a little bit, let's see here. Yep, you see that, the SH? So when you have those two amino acids close to each other due to the tertiary, formation, they form this disulfide bridge. You see that? Okay, so you can see how all of these bonds will create this three-dimensional tertiary stabilization. Alright, so now we're going to just go over the different protein structures again, from the simpler one to the complex ones, okay? So primary structure is basically like it says, right? It's a primary single simple amino acid chains linked together, okay? So now, when this simple, amino acid chain is kind of starts foiling and folding right on each other, those amino acids have basically side chains. Remember, those side chains will result in hydrogen bonds between repeating polypeptide backbones. Okay, so they can form alpha helix. Okay, when they form coils. So when they're like around, they form like this around each other. That chain. We have alpha helix that's being formed. Okay. We have another also secondary structure, and that would be the beta plated sheet. Okay. Let me go back a little bit to show you what that looks like. You see that? That's when we're talking about secondary structure. That would be your coil. You see that hydrogen bond I was talking about? Because they get close to each other, that hydrogen bonds form. And when we talk about beta plated sheets, they're basically sheets, okay, kind of like that. And when they get closer to each other, you can see how they form these hydrogen bonds as well. Okay. So let's talk about the tertiary now. Tertiary structure. Okay. Tertiary again has several types of bonds, right? Depending on the side chains of amino acids, right? And then the R groups. Okay. So they can be, hydrogen bonds. They can form ionic bonds, hydrophobic bonds, such as van der Waal, right? They can form this disulfide bridge, as we talked about. Okay, these are actually strong covalent bonds. Okay, and they maintain structure of that protein. So let's talk about the fourth kind. This would be quaternary protein structure. And this results from two or more polypeptide chains. Okay? Collagen is basically an example of that, and that's a fibrous protein. Okay? It is formed by three polypeptide coiled like a rope. You guys can watch this on your own. Okay? Hemoglobin is another quaternary protein structure. Okay, and it consists of two alpha and two beta subunits of polypeptide chains. And hemoglobin, as you know, or you might know, is what allow the cells to carry oxygen. So you can imagine how important this protein structure and its formation. Okay. And this is a actually depiction of hemoglobin here. You can see these are red blood cells. Okay, you can see that there is two alpha subunits. You see them in the dark blue, and then two beta subunits. Okay, and we can see how this hemoglobin. You see it here. That's the whole thing. Hemoglobin. I mean the heme, right? Is actually allowing it to carry iron. Okay, and this whole thing will allow the body to carry oxygen all the way to the rest of the body. So sometimes some patients have basically a problem. Some people have a problem with this. The formation of red blood cell, okay? And when this happens, they have a sickle cell or sickle cell's disease, okay? You're seeing a normal blood cell, red blood cell here on the top. You're seeing a sickle cell on the bottom, okay? This results from a slight change in the protein structure, okay? It's actually an inherited blood disorder, so it's basically passed on from parents. Okay, and what happened is that there's a single substitution of amino acid here. You see those amino acids? That's the chain we were talking about. And when it's substituted by another one, just a simple substitution, there is a defect in the formation of red blood cell as a result, and you can see the beta subunits and alpha subunits how they get affected tremendously because of that. And due to all of this, instead of them being able to carry oxygen individually and not aggregate as they're not supposed to aggregate, you know, because of that uh, basically defect, they start aggregating, as you can see here. And that aggregation, what it does is it blocks um, the blood from flowing in arteries and so forth where the blood goes. Okay, you can imagine the loss of blood to a certain body part is gonna basically deprive it from oxygen. Okay. Okay, so we're gonna talk about protein denaturization. Okay, so that's when we basically change the shape of a protein structure, and this is basically have a tremendous effect on the function of that protein. Okay, we call this denaturization. Okay, so denaturation. So this results from an alteration in pH. Okay, so when we change due to an acidic environment or a basic environment due to salt concentration, obviously temperature changes, and other things. Okay, a denatured protein is basically biologically inactive. Okay, in the case of fever, right, we have proteins in the blood basically tend to denature at very high body temperature. You could imagine this is not a good thing. Okay, sometimes it can be reversed. Okay, but sometimes it's not always possible. Another example of denaturation is when you um, look at an egg, right? An egg, when you crack it open, it's actually in the liquid form, right? Transparent liquid form. But whenever um, you try to cook it, right? This transparent liquid protein becomes white and becomes solid as well. So you can see that it changes the structure of that protein. Okay, and even after you cook that egg and you leave it outside or whatever, it's not gonna go back to the liquid form anymore. Okay. So just know how important the protein structure is, right? There are several diseases like Alzheimer's, Parkinson's, and mad cow disease. They're all associated with misfolded proteins. Okay. How do scientists, know about the structure of protein, which is important? They use, X-ray crystallography. Okay, that's actually when they whatever they're trying to find basically, they hit it with X-ray beam, and that X-ray beam basically diffract, um, using a crystal here, and that diffraction basically will allow them to see the shape of that molecule directly, right, and the location of the different atoms of that protein. Okay, another method would be nuclear magnetic resonance spectroscopy. Okay, bioinformatics is basically the study of allowing um, scientists to use and study different protein structures and amino acid sequences. Okay, so they use computers specifically. Scientists use computers specifically in bioinformatics to study and research. I'm talking about thousands and millions of protein bases and amino acid sequences and DNA sequences as well. So we're gonna talk about nucleic acids as the final thing for this chapter. So nucleic acids are basically, what make up the inheritance, in a gene. Okay, so they, they're the ones that form our DNA. Okay, and our DNA are made up of basically the monomer um, form of these nucleic acids, which we call nucleotides. Okay, so several nucleotides make up our DNA. Okay, there are two types of nucleic acids. We have something called deoxyribonucleic acid. That's DNA. That's what our cells use as the coding um, information for everything. There's another type that's called ribonucleic acid, RNA. Okay, we'll talk about what the difference is. Again, DNA provides replications, right, for directions for its own replication, as well as directions for pretty much everything else in the body. Okay, it also directs the synthesis of messenger RNA, or what we call mRNA, and basically that's what controls protein synthesis. And we will see how that all that process happens in more detail. All right, this process of going from DNA or gene to a protein, we call this gene expression. All right, this is a simple depiction of how all that process happens in the cell. So you can imagine this whole thing is a cell, and this round part is the nucleus of a cell. Inside the nucleus, you will see the double helix DNA, and basically what happened is this DNA um, basically transcribes the messenger mRNA. That's how it works in that specific order. After mRNA is made, um, this mRNA or messenger RNA leaves the nucleus, as you can see here, and inside that cytoplasm of the cell or the environment around the nucleus, it basically uses ribosomes, which is another structure in the cell, to synthesize protein or polypeptide chain. Um, and this is how you get your amino acids. Okay, this process from mRNA to protein, we call this translation. And we will talk about all of this in future chapters in more detail. Okay, so we call nucleic acids polynucleotides. Okay, so the nucleotide is the simplest monomer, and when we have several nucleotides attached to each other, we call this a polynucleotides. So each nucleotide or each monomer, okay, it consists of a nitrogenous base, a pentose sugar, and one or more phosphate groups. We will see that in a little depiction in a bit. The portion of a nucleotide without the phosphate group is basically called a nucleoside. Okay, I know they're close terms, but that's what we call them in science. Okay, so let's take a quick look here at this nucleic acid here, the polynucleotide here on the left side. You can see that this that's actually a zoomed in to let you guys see how that actually looks like at close up. So the yellow circle is your phosphate group. The green pentose ring, that would be your the sugar or the backbone, and the you have your nucleoside or that would be your nitrogenous base. Okay. You can notice that it starts at the fifth end, and it ends on the third end. Okay. So what you will see is that we have two types of nitrogenous bases. Okay, and that's important, right? We have pyrimidines and purines. Now, pyrimidines consist of cytosine, thymine, and uracil. Okay, you can see the differences here. Cytosine, thymine, and uracil. Purines are adenine and guanine. Okay, what you will notice is that purines are basically a six-membered ring fused to a five-membered ring. You can see that. See that? The pyrimidines have just a single six-membered ring. Okay. Notice here that uracil is actually a nitrogenous base that's actually only used in RNA. Thymine is only used in DNA formation. We will go into deeper study in a bit of what we're talking about here. I know this is new information. Okay. Now, as far as the sugars, we have deoxyribose in DNA and ribose in RNA, right? That's why we call deoxyribose nucleic acid for DNA. We call it ribose nucleic acid for RNA. Okay. The difference is this hydroxide group here. Okay. You'll see there's a loss of hydroxide in DNA here. All right, so you guys look at this animation on your own. So nucleotides are linked together by a phosphodiester linkage to build a polynucleotide. Let me see if there's, we didn't include one for you. So that phosphodiester linkage is basically between the different nucleotides, okay? Just remember that this is formed in between nucleotides. Okay? And a phosphodiester linkage consists of a phosphate group that links the sugars of two nucleotides. See that? So it actually acts on the sugars, and that when we say the sugar is this part. You see that? It basically links those. You can imagine this and this. If we draw a line in between here, that would be our phosphodiester linkage. So these links create the backbone of sugar phosphate units, right? With nitrogenous bases as appendages. Okay, so these nitrogenous bases that, in the case of DNA or RNA, right, these will be basically facing the inside of DNA. DNA, as you know, is made out of double helix, so there's two chains that are running together. Okay. What happened is when these face inside these nitrogenous base, they bond with each other, right? And that's how we have these nitrogenous bases bond with each other. So in the case of DNA, cytosine normally bonds to guanine, and adenine bonds to thymine normally in DNA. Okay. Now in uracil, adenine will bind to uracil in RNA. Okay, because RNA does not have thymine. So again, as I mentioned, DNA molecules have two polynucleotides spiraling around, right? And this is what we call the double helix. Okay, the backbone run in opposite 5 to 3 direction. Okay, so one runs 5 to 3 this way, and the other one will run 5 to 3 this way. Okay, and that anti-parallel direction is what allows the DNA molecule to twist around like that, around itself, and allows it to have those bases, nitrogenous base, bond with each other. Okay, so only certain bases of DNA pair up. As I mentioned before, the adenine binds with what? Thymine. In the case of DNA, guanine binds with cytosine. So just remember, G in guanine binds with C in cytosine. A in adenine binds with T in thymine. This is what we call complementary base pairing. Okay, these bases have to pair and are in pairing mode um, inside a double helix DNA. Okay, just remember that the bonds that is formed between the bases, so between, let's say, adenine and thymine, they're actually hydrogen bonds. Okay, hydrogen bonds. However, the bonds that are being formed between the different nucleotides, right, between a nucleotide and another, between the sugars of both nucleotides, is what? Phosphodiester linkage. So this is the difference. Phosphodiester linkage is between the two nucleotides, okay? And each nucleotide has what? A sugar and that nitrogenous base that we're talking about, which is, it could be adenine, it could be guanine, whatever, right? One of those 4 bases, okay? So this complementary base pairing is a feature of DNA that makes it possible to generate basically 2 identical copies of each DNA molecule. This happens when basically the cell prepares for division, or to make a copy of itself. All right. So we will learn about how that process happens in a bit in future chapters. Now, when we talk about RNA, the ribonucleic acid, okay, ribose nucleic acid, that's basically single-stranded, so it does not have a double helix. In the case of RNA, it still has like complementary base pairing, and the reason is because although it doesn't have a double helix, okay, where there's like 2 chains wrapping around each other and their bases pair, the reason why it still has complementary pairing is because RNA basically wraps around each other, and its nitrogenous bases also form those complementary bases as well. Remember that in RNA, basically the thymine is replaced by uracil. Okay, so uracil we depict it with a U. Okay, so adenine and uracil bind together. Okay, and this is a depiction of the differences between the DNA structure. You can see the double helix. You can see the RNA structure. It's just a single, and it goes around it, itself, forming those bonding in between the bases. You can see how the bond between the bases is what? The nitrogen bases. They're hydrogen bonds. Okay. Now this is the focus on the blue part. The blue part is your backbone. Your backbone is made out of what? A sugar and a phosphate. Remember that? What allows these sugar phosphate nucleotides to stick together and form that ribbon-like structure? Phosphodiester linkages. Remember that? Okay. So once the structure of the DNA and its relationship to amino acid is understood and was understood for several years now, biologists have been able to decode genes, right, by learning their base sequences. So they can basically learn much more about the gene and its ability to code for something in the body, and whether it's a function, whether it's a structure, and sometimes a defect in that specific gene is in what we call mutation, will basically generate a disease. Okay. So the scientists have been able to actually um, sequence the whole organism's genome, and they call this the Human Genome Project. So they were able to basically sequence about 3 billion uh, genes in the human body completely. Okay, so they were able to basically see all those bases. Remember those nitrogen-based bases? The identification of those bases is basically what allowed scientists to know about the different uh, genes and how they form a specific DNA sequence. Okay. Again, we talked about bioinformatics. So bioinformatics uses computer software and computational tools to collect those data sequences from many genomes. Okay. Analyzing large sets of genes or even comparing whole genomes of different species is called genomics, as the name states. Okay? Genomics for large sets of genes. Proteomics is basically analysis of large sets of proteins. So you can see the name match the thing you're trying to study and analyze. Okay? And the next few slides are just a review of the things we talked about already. I'm not going to go into detail right now anymore because we already did. Just this is a summary for you to basically focus on and the study guide for this chapter. Okay. All right. So that ends our lecture for today. And with that, I will see you all probably next time um, in chapter 6. Have a good one, everyone.